Hello, my name is Jaqueline Quintanilla, and today I will be presenting about my project, the application of PLGA Microparticles with targeting agents for the treatment of glioblastoma. So each year, around 10,000 patients in the US are diagnosed with glioblastoma multiforme, or as we call it GBM. And despite the treatment options already currently being used, the survival is only about 21 months after diagnosis. And GBM also expresses various antigenic properties that help it in its progression and evasiveness. Therefore, we try to find a new form of treatment for this deadly disease. Minocycline is our main focus and the drug that we use. It is a tetracycline antibiotic that has been used for other infections, but it's also shown to inhibit U87 glioblastoma cell death and also inhibit angiogenesis. In this project, we'll also be focusing on CD44, which is a receptor found to be highly expressed in glioma cells compared to other regular cells. And it has also been correlated with the more severe and aggressive forms of GBM. For this project, in order to conjugate our microparticles with the anti-CD44 antibody, we're going to be using the carbodiamide chemistry, which we found through literature research to be one of the most preferred and effective manners to conjugate antibodies to specifically PLGA microparticles. So as shown in figure 1, you can see that the chemical structure change that happens when through the carbodiamide chemistry, if it effectively changing the PLGA structure to an amide with the antibody. So our hypothesis was that the conjugated PLGA microparticles will be more effective in targeting glioma cells than the unconjugated microparticles. And the overall objective of this study is to develop minocycline-loaded PLGA microparticles and conjugate them with the anti-CD44 antibody to directly target glioblastoma cells in order to better treat GBM. So for the methods, one of the most important parts is the antibody conjugation process. In this process, we dilute 10 mg of our particles that we make in the lab with 1 ml of 0.1 MES buffer, 100 microliters of 0.1 molar EDC, and 100 microliters of 0.7 molar NHS. So we just shake this solution for 1 hour at room temperature, and after that we centrifuge at a high speed of 21,000 times 3 for 10 minutes at 4 degrees Celsius. After that, we get the solution, we dilute again in 10 ml of PBS, and at that we just use 1 ml of the solution and add the 10 microliters of the antibody. After that, we centrifuge at the same speed, with the same time and temperature, and we wash 3 times with PBS, and making sure to save the supernatant for our future study, which is the antibody conjugation efficiency using the Bradford assay method. So for our results, we found that the antibody conjugation efficiency was very good. It was at 70.8, almost at 70.8% through all the 3 trials. So as you can see in figure 2A and B, you can see that each trial had 4 different washes and each with 3 different samples. So we have the overnight, which is basically the one that stays the whole 24 hours incubation with the antibody and the microparticles. So as you can see, that one does have the highest antibody concentration. So it has the highest absorbance, if you can see in figure 2B, which is why it has a darker blue color. But each wash, there's going to be less and less antibody present, which is why there's a least absorbance. And also overall, when we total the 4 washes, we have a much higher conjugation efficiency since there's a smaller amount of absorbance. So we had a 70.8% average efficiency, and that makes us know that this carbodiamide chemistry was effective in conjugating our anti-CD44 antibodies that we wanted to use with our specific microparticles that we made in our lab. So we also looked at the morphology of the microparticles before and after conjugation with the carbodiamide chemistry protocol. We can see in figure 3A that the unconjugated microparticles did have like a spherical and smooth appearance. So that is good to know because that is essentially what we want to have. And then in figure 3B, we can see that even after the conjugation, they also have that same or similar structure of a sphere. So we know that it didn't really change. If there wasn't any drastic changes in the structure and morphology, which is good. And we could also see that there is only a slight change on the surface of the microparticles. So they become a little bit more texturized, as you can see in figure 3B, those two pictures at the bottom. You can see that the structure has a little bit of like a texture, like little bumps on the surface, which we think might be caused by the antibody conjugating to the surface of the PLGA. But it is also very important just to know that the structure overall didn't change, and the conjugation efficiency was very high at 70.8%. So this just makes us assume that this carbodynamic chemistry was effective for ours. So in conclusion, we can see that the PLGA microparticles that we fabricated through the use of electrospraying were successfully conjugated with the antibody of our choice, which in this case was the anti-CD44. At a high conjugation efficiency using the carbodynamic chemistry of EDC and NHS, while also keeping the overall spherical morphology. So in the future, we also want to see how these microparticles are going to be uptaken by the cells of our choice, which in this case, we're going to be using the U87, which are very important to represent the GBM that we want to treat. So the interaction of the conjugated microparticles to U87 glioblastoma cells will be determined using the light and fluorescent microscopy to determine the overall effectiveness of the conjugated microparticles compared to the unconjugated ones that we also have. So that is it for the project. We have the references, and also I want to acknowledge the Engaged Scholar and Artist Awards for their funding on the project, the Biomedical Science Program at UTRGV, and my research mentor, Dr. Silverio, and her love for the continued support and guidance on this project. Thank you.